The very first PowerPoint that we're going to go through for Unit 3 Biology and the very first content area we're going to look at is all about plasma membranes, and hopefully you recognise that plasma membranes is something we talked about quite a bit in Unit 1 Bio, and so most of the stuff that we're actually going to talk today is going to be a revision, but we're just going to go into a little bit more depth, right? So in your study design, you have to know what this fluid mosaic model is of the plasma membranes, and how the movement of what we call hydrophilic and hydrophobic substances move across that plasma membrane based on their size and based on their polarity. Before we go on, these two words, hydrophilic and hydrophobic, are very, very important, and hopefully you can recognize that hydro, we're talking about water, right? And then phobic, hopefully you can see, means scared or dislikes. So hydrophobic means dislikes water. Philic means loving or likes. So hydrophilic means water loving. So why do we study this organelle, the plasma membrane? Why is it the first part? You'd think, let's study something as the first part of Unit 3 Bio. You'd think we'd study something like the nucleus, the power, the center, the brain of a cell. But we study the most outside part that every single cell has, this plasma membrane. And the reason why is that because the plasma membrane is the gatekeeper of the cell. All right, nothing comes in, nothing comes out without the plasma membrane. Okay, and this actually means that Unit 3 Bio is actually centered on the plasma membrane. In Unit 3 Biology, remember, we're going to talk about things like communication, which happens via the plasma membrane. All right, we're going to talk about exchange of things during cellular respiration and photosynthesis, and that exchange happens via the plasma membrane. So really, the plasma membrane is a control for what's happening to the cell. Okay, it's the first line of the cell. So let's talk about the structure of the plasma membrane. So the plasma membrane, it is a structural organelle. Okay, it is an organelle, and it encloses all of the contents of a cell, acting as the division between what we call the intracellular and the extracellular space. So intra means inside the cell, extra means, exit, outside of the cell. Okay, so it's a 7 to 9 nanometer thick phospholipid bilayer with associated proteins, carbohydrates, and cholesterols, which is a lipid or a fat. Okay, and we call this the fluid mosaic model. So if you look here, we've got this yellow and green part, that's the phospholipid bilayer, and then inside we've got things like proteins, we've got carbohydrate chains attached to more proteins, and we might even have cholesterol infused into the membrane. So why is it the fluid mosaic model? Because just like this membrane here is showing, it can move, right? It's very fluid, all right? And it's a mosaic because it's got all these things embedded into it. So these proteins and carbohydrate chains, and we've got these cholesterols embedded inside, all right? But we might look at the membrane one day and it'll look like this, and then we'll come by to the membrane the next day, and this, for example, transport protein might have moved all the way down there, and this one might have moved down here, right? So there's a big fluidity of the membrane. We can actually just completely, and that's the reason why cells can completely move around, and anything we need on one side of the cell, we might need later on the other side of the cell, so those things can move within the membrane. So that's why we call it the fluid mosaic model. Alright, so let's talk about the structure of these phospholipids. What phospholipids are, is a hydrophobic fatty acid tail. So that's this part here. Alright, so that's hydrophobic, which remember means scared of water, right? And it's attached to a hydrophilic phosphate phosphate head. That should say phosphate. Tiggy. And so this phospholipid bilayer, it naturally undergoes this structure that we often show like this for the membrane. And the reason for this is because our bodies, remember, are 70% water. All right. So outside here in the extracellular space is full of water. Outside here in the intracellular space is full of water. So naturally the plasma membrane organizes itself like this. Okay. So if you actually just threw a bunch of phospholipids into some water, they would naturally get this structure. So let's talk about some of the functions of this plasma membrane, and this little Giphy is really great at showing the fluid mosaic model. As you can see, it's moving around and all of these things that are inside of it can easily move around. But so the main function of the plasma membrane is its selective control of molecules entering and leaving cells. We call this a semi-permeability. So it's not fully permeable, okay? It's somewhat permeable, and it selects what molecules comes in and out. Also, the plasma membrane is very important for protecting and separating the cell, so the intracellular environment, from its surrounding, the extracellular environment. Right? The fluid structure allows for flexibility and changing of shape with cholesterol intertwined in the tail. So this cholesterol, these fats, actually allow the flexibility and resists temperature changes in the membranes. And we're going to run a little prac at the start of next year on this. So the plasma membrane, it has these proteins that are inside of it. So the next most important thing after the phospholipid are these proteins, right? And these proteins can be categorized into 4 types. The first type is the first one we're really going to talk about. We're going to start talking about them today, is the transport proteins. And they can be either channel proteins or carrier proteins. And what they do is they assist the movement of molecules which can't simply cross the semi-permeable membrane. So lots of things can actually just go straight through the phospholipids, but if it's too big or its charge is wrong, right, it's actually going to go through one of these transport proteins if the cell's going to need it. Another very important component of the plasma membrane are receptor proteins, and what receptor proteins do is they receive molecules, which we call ligands, from the extracellular space, which communicate with the cell. Right? So if you imagine a molecule might come and attach itself to one of these receptor proteins, and that will give them the cell a message or something. And then recognition proteins, they're very much like receptor proteins, but they're actually important because they actually interact with molecules, cells, and other cellular agents and identify whether it's native or foreign. So, for example, if something meets up with a recognition protein, right, that recognition protein is going to say, oh, okay, that's just another one of the cells of Mr. Mallow's body. Or it's going to look at this recognition protein and go, oh, this is foreign. I'm going to have to attack this cell later on. And then lastly, a function that we're not going to talk very much about is just the adhesion function of proteins for cells. So for example, this protein here, if this was skin cells, we want the next skin cell to be right nice and attached here. So this adhesion protein would adhere to the cell that's next door. All right, and that makes sense. You don't want to be able to put your finger through your skin, and that's the reason why we can't. So let's talk about the first kind of proteins first, and transport as in general. All right. So different substances can actually move across the phospholipid bilayer. Okay, and what decides whether they can or not is their size and their charge. Okay, so let's actually start up here. So hydrophobic molecules, so these are non-polar molecules. They can just naturally just pass through the membrane, especially if they're quite small. For example, oxygen, carbon dioxide, a nitrogen gas. They just go and flow through the membrane according to the concentration gradient. All right. Some small uncharged polar molecules, right, so these are polar, if they're very, very small like water, especially like water, but indoles and glycerol, for example, as well, they can also naturally pass through the phospholipid bilayer. Okay, and water we're going to talk about in a second, quite a bit about this. two different kind of molecules aren't going to have a very easy time passing through the phospholipid bilayer. The first kind is if the molecule's really, really big. Right? If it's too big, right, especially if it's too big and it's polar, right, it's not going to just pass through the membrane. And a really good example of this are the carbohydrates or proteins. Okay, they don't just run through the membrane. Okay, they're going to need a transport protein there. And then lastly, ions, all right, because ions have this charge next to them, this negative or this positive charge, and that doesn't allow them to pass through the membrane. Why? Why doesn't it allow them to pass through membranes? Because of what we call polarity. All right, and polarity is all about water. So water is a molecule that is H2O. So it's one oxygen and two hydrogen atoms together, all right? And H2O has what we call polarity. That means that on this side, H2O has a slightly negative charge, and on this side, H2O has a slightly positive charge. What does that mean? When we have the phospholipid bilayer, let's draw a phospholipid bilayer here. Remember that the outside of the bilayer is hydrophilic. It loves water. That's why it's facing out. But this inside part of the bilayer is hydrophobic. So it hates water. So whenever a molecule which is polar, so for example, an ion, chlorine ion, tries to get through, it can get to here, but then it's sort of stopped. It's blocked here because this part is hydrophobic, okay? But an ion, if it were to meet a water molecule, it really likes that water molecule, and that's the reason why salt, NaCl, dissolves in water, because ions really like water molecules, right? And that's the same with any polar molecule. So for example, if we were to have an amino acid, right, and this amino acid had some kind of charge, or it was simply polar, so it had one side that was positive, another side that was negative, it would be loving this water, right, because it's hydrophilic, because it's polar. But if it tries to go through the membrane, all right, it would once again get stopped here. Okay, and that's what polarity means, especially for biology. That's all you have to know about polarity. All right, let's talk about the different types of transport. All right, and there's four major groupings of transport. The first three types of transport are all what we call passive transport, which means that there's no energy needed, there's no ATP needed. Okay, so the first kind is simple diffusion. Well, the first two kinds are actually fall into the simple diffusion category, and that's any small, uncharged, non-polar or hydrophobic molecule is going to naturally just pass through the plasma membrane unassisted, as long as the concentration gradient allows for it. Okay, so that's this kind of molecule here, and this might have been an oxygen molecule, for example. Or what this can actually be is a water molecule. Right. Although water is polar, it's so tiny that it can still cross relatively unassisted. And we call this special kind of simple diffusion, we call this osmosis. Okay. And then the other kind of passive diffusion is called facilitated diffusion. So simple diffusion is just happens. Facilitated comes from the word facilitate, which just means to help, and that just means that a large charged polar or hydrophilic molecule can sometimes be assisted through the membrane via these transport proteins like this. So here we have a channel transport protein, and here we have a carrier transport protein, and what these are going to do is allow these molecules that shouldn't usually pass through the membrane just to pass through. As long as the concentration gradient is being followed, this isn't going to require energy still. So what is this concentration gradient? It's the chemical driving force that pushes molecules into and out of cells. Okay, so for example, here the concentration gradient is very high inside the cell, very low outside the cell. So naturally the molecules are going to go out. Here we have the other way, low inside the cell, very high outside the cell. So the molecules are just going to go in. Okay, this kind of concentration gradient being followed, we're talking about passive So no energy required. But what if we want, for example, this molecule to get out? It's a bad molecule. The cell doesn't want it. So we want this molecule out. So we're going to go against the concentration gradient. And that's what we call active transport. Okay? And how this is done is by transport proteins that just grab that molecule, and we provide some ATP, and that ATP forces those transport proteins to push those molecules against the concentration gradient. Okay? So that's pretty much plasma membranes. Make sure you answer these 3 quick questions on the Google Forms that's on the website, and we'll go through them in class together.